Okay, guys, so these are your notes for DNA history and replication, part 1 and part 2. And I've got 2 of your classmates with me, so they're going to be helping ask questions as we go along. I don't know how to change the slide. Hopefully I didn't do weird stuff. Okay, so on the first slide, you guys see, and can you two see? You see that you have Alfred Hershey and Martha Chase, and the only thing that's important to know about them is that Hershey and Chase are largely responsible for finding bacteria, or for finding DNA and knowing what DNA is. So they're not just showing you a picture like because they want you to think they're married or something crazy like that. These are just the 2 people that we credit as being responsible for figuring out what DNA looked like and all that fun stuff. And so what happened is they worked with a bacteriophage, which is just a type of virus that infects bacteria. But remember that everything that has everything that's alive has DNA in it, right? So it doesn't matter if you're a prokaryote or a eukaryote. If it's alive, it's got DNA in it. And so this slide is just showing you the virus, which is double tap draw shape, which is I don't does that show up there? Well, I don't know how to change the color. Do you see how to change the color?
Nope.
No, it's just gonna make right there. Okay, so these big old things that you see are just your viruses, and it's showing you the 3 different parts to the virus, which are going to be your head is this thing, if I can get this stupid pointer to work. Your tail is this area, tail fibers, and then down here you have the cell's DNA. And so what's happening is that virus is infecting the bacteria, and these are the same things, like the same viruses you guys get. So you know that you get like a flu shot, so that way you don't get the flu virus. So all the flu virus is this weird looking doohickey, and the issue there is that viruses are like, they're alive, but they're not alive, which I know doesn't make a lot of sense. But you can't kill them because they're not alive. Does this make sense? Mm-hmm. And so that's why they give you the vaccine for the virus, is so that way you don't catch it in the first place. Because once you have the virus, there's really nothing that can be done for the virus. You just have to wait until it goes away. But what they did was they worked with this bacteriophage, this virus that infects bacteria. And then remember, if you flip back to the very first unit when we took our notes, your electron microscope is just that microscope that's going to scan over and kind of give you a 3D image. And so what it's showing you is all of these little nasty bubbles that you see going on are all viruses. So this whole entire thing, this little, we'll make him blue. This thing is your bacteria. And all of these guys are your viruses that have infected the bacteria. And you see that they're just all over the place. All right, so let's look back at this picture. And what we see is the sulfur, you remember, was pink. Yeah. And the phosphorus was blue. So if I'm mixing the sulfur that's radioactive pink with bacteria, and it's not going inside the bacteria, how do I know that it's not inside the bacteria?
You see the protein. You will see it.
I would see what?
The protein shell. Not the protein shell, but
you would see it sitting down at the bottom.
Not necessarily sitting down at the bottom, but look, okay, so let's say that all the bacteria are in this pellet, right? What do you notice about the different colors of the 2 pellets in there?
One's more than the other one.
One's more what?
One's more full, and it's like higher.
That's not what I asked you. What'd I ask you?
Well, what's the difference?
So what Hershey and Chase did is now known as your Hershey-Chase experiment. And so it says the first thing that they did was they mixed radioactively labeled phages with bacteria, and the phages then infected the bacteria. Then they agitated it, and all that means is they shook it up in the blender to separate out the phages outside the bacteria from the cells and their contents. They centrifuge the mixture, which is just where you spin it down, so that way it separates the different parts. So everything that's really heavy is going to get sucked to the bottom, while everything that's not heavy is going to float at the top. And that's what a centrifuge does, and that's what it's showing you in this picture, is that the pellet is this thing down at the bottom. That's that heavy section that's been pulled down, which means that the radioactivity in the liquid is going to be pulled up to the top, so they can measure how much is in the pellet and how much is in the liquid, because now they're two separate things. And they did this with two different elements. They did it with sulfur and they did it with phosphorus. So if you look at your notes, it says they used radioactive sulfur 34 to label the virus's protein. Then they used phosphorus to label the DNA inside the virus in another container. The radioactive viruses were then exposed to bacteria, and the virus infected the bacteria. So this goes back to when we were talking about photosynthesis, right? And I said that if we made the oxygen on the water glow in the dark. Okay, so what I wanted you to see there was, what do you notice about the two different colors of the pellet?
Is that empty at the bottom?
What are the different colors of the pellet?
Oh, blue
and pink.
One of the pellets is pink.
No,
one of them is blue, and then the other one is like gray. Or
The other one's gray. So we're saying that that sulfur did not mix with the bacteria. So you notice that the pellet where the bacteria is not pink. Our virus is pink. If our virus was in the bacteria, then the pellet would also be pink. Does that make sense? As compared to down here, where we see the phosphorus did match up and infect our bacteria, down here our pellet is what color?
blue.
Blue, just like the blue virus. Does that make sense? Okay, so that's what it's talking about, where it says that in the radioactive phosphorus container, the radioactive phosphorus did enter the bacteria. When they were reproduced inside the bacteria, the reproduced virus that came out of dead bacteria were radioactive. So what they proved was that DNA was a transforming agent, and that it carries the information blueprint from one generation to the next. So what it's talking about is something that we already know, right? We know that DNA is passed down from one generation to the next, whether that's in a bacteria, which is going through binary fission, or we know we're going to start with, I don't know what I just did. Where we know we're going to start with one cell who's going to split into two cells, but these two new cells are going to be exactly identical to this first cell. So when they split into two new cells, these four cells are going to be the same genetically as that one, right? And we know that's for bacteria. But where else do we see DNA passed down?
Plants.
In plants. Where else?
animals.
In animals and humans, right? That's why you might have your mom's eyes or your dad's nose or whatever, right? Because you're getting their genetic information. Okay, so this is Erwin Chargaff, and now we're on Roman numeral 2. And he developed what is known as Chargaff's rule. And the rule states that for all organisms, A equals T and C equals G. So all this is talking about are your base pairings, which is the way that they line up in DNA. And so we've talked about this before. You kind of already should have an idea on this, which this is just your cytosine is going to pair up with guanine. Your thymine is going to pair up with adenine. Remember that we made our own DNA inside the classroom. Some people had fists, some people had hands. You had to match up a hand to a hand, a fist to a fist. And then you had the short arms had to match up with the long arms. Do you remember this? Yes. With your purines and your pyrimidines? Okay, so that's all basically from this guy. And so that's exactly what it shows you there. Adenine and thymine pair up when you're looking at DNA, but in RNA, instead of having thymine, you have uracil. So that U is going to pair up with your adenine. It's going to pair up with your A. So you kind of need to go ahead and memorize that, which is just that in RNA, you're going to have uracil instead of thymine. And in DNA, you have thymine instead of uracil. But regardless, you're going to have adenine. Adenine is always there, guanine is always there, cytosine is always there. And so what this is telling you is that, thank you, ma'am. What this is telling you is that if I have 20% adenine, what percent am I going to have thymine? If they're equal.
20%.
20%, right? So let's do some math here. If we said 20% adenine has to be equal to 20% thymine, right? What's 20 plus 20?
40.
What's 100% minus 40%?
60.
So that tells us that this remaining amount of guanine and cytosine has to be equal to 60%. Now they're also equal to each other. So how can I figure out how much guanine and cytosine I have?
Add them together.
Add what together?
60% and 40%.
No, 60% and 40% gives me 100.
60% and 20%.
Mm-mm. 60 and 60 make 120. You're trying to figure out guanine and cytosine are equal to 60%. How can you figure out how much each one of them has? Divide by 2. So 60% divided by 2 means that you have 30% guanine and 30% cytosine. And so this is a pretty big deal, so make sure that you underline it, make sure you write it in bold, make sure you circle it, but that's what it's showing you under number 1. So it says, for example, if you know a species has 32% thymine, there's some kind of way, oh, what the, I don't like that. Oh, okay, I don't know if that did a good thing or not. Okay, I think I just made a new slide. So it says that you have a species. So we'll say it's a frog. Our frog, we're looking at his DNA. So what base pairs am I going to have if we're looking at DNA? What letters? We are looking at DNA. So you should right off the bat be able to tell me 3 letters because there are 3 of those letters that don't ever change. All right, ladies, that's your grade you're playing with. You always have adenine, you always have guanine, you always have cytosine, and because we're looking at DNA, we know that it's going to be thymine. And so what this problem is telling you is you have 32% thymine. Now thymine and adenine are always going to pair up. So let's do them in black, but let's change the letter for guanine and cytosine to blue. So adenine and thymine always pair up. So if you have 32 T's, how many A's do you have?
32.
So you're going to have 32% A, 32% adenine. Now what's 32 plus 32?
64.
All right, make sure you're not just reading that paper. You need to understand how to do it. 64. And so since we know the total is going to be 100, 100 minus 64, 64 minus 100, doesn't matter to me, is going to be what? 36. So that means that the remaining pairs, our guanine and our cytosine, have to equal up to 36% total. So to figure out how many guanine and cytosine we have, we divide it by 2. 1 for guanine, 1 for cytosine. And that gives us what? 18. So that tells us we have 18% guanine. And we have 18% cytosine. And that is the ugliest G I've ever drawn in my life. There we go. And so that's what it's showing you when you look at your, when you look at that example there under Chargaff's name. So, we're going to do a couple more of these during class. And if I'm telling you that now, what do you think that means?
Study.
That you need to study this between now and tomorrow to make sure that you understand it, to make sure that you can figure it out.
Can I get my test and the rest of my work? Because I can't come back to school for the rest of the week.
You can right now. We're recording the lecture, so hold on for a second, okay? So this is Rosalind Franklin, and she decided to come out and play in the 50s. And so what she did was she did an X-ray crystallography on DNA. And what that means is the picture, she made the picture of DNA. She helped them figure out what DNA looked like by doing an X-ray of it. And so let's go ahead and look at that slide. And so she X-rayed the DNA molecule, and it showed her that there is a double helix. So what does the word double mean?
2.
2. Do you know what a helix is?
Isn't it the thing that makes the little...
Exactly right, Julia. That is the little doohickey that makes our DNA look like it kind of spirals back and forth. Come on, Pen. Right? So that's our double helix. And so that's what this picture is showing her, is that you have it crossing over and making kind of an X inside of our X-ray. And so from a side view, this is what we see. What are this T and A, do you think?
Thymine and adenine.
Thymine and adenine. And our G and our C are going to be
Guanine.
guanine and cytosine. And then hopefully you can kind of recognize what's going on with the rest of these. If not, you really need to be studying your notes on those macromolecules. So we see here that we have this doohickey, which we've talked about a whole bunch before. You should be able to recognize that as being a sugar. Then we have this other thing that has a P in the middle, which we should be able to recognize as a phosphate group. And so if you don't remember that, you need to look back over your nucleotides, macromolecules notes and make sure that you understand what DNA looks like, what DNA is made of. Now, this is still showing you the double helix that Julia mentioned just a second ago. And you can see that right here, which is how you're going to see it a lot in your textbooks. It's not going to be quite like that first one I drew. And you can also see it over here, right? So this is giving you more of a 3D image. So let's make this a cool 3D outline. Which is why you can only see the bands sometimes, because they're going to overlap each other. But what you should notice on all of them is that they're all showing you the same thing, which is that you have adenine and thymine are going to pair up. Adenine and thymine, 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 adenine and thymine. And you have guanine and cytosine, which are going to pair up. Guanine, cytosine, cytosine, guanine. And it doesn't matter what order they pair up in, so long as they always pair up with their partner. Right? Does this make sense? Yes. Kind of like if you think about you and Bay. It doesn't matter where you and Bay go on a date with, as long as it's you and Bay. The day that Bay decides to pair up with somebody else is going to be a problem, right? Yes. So it's the same thing with your DNA here, is they always go with their dance partner. And so let's look at our notes. So it says the picture indicates a double helix. This picture would be from the view of looking down at a strand of DNA. So that's talking about that slide we saw before, right? And so if we're looking down at one of these pictures from the top, it's just going to look like an X, right? And then you're going to be able to see kind of So what you're going to be able to see is kind of a spirally motion going down here. But if you're just looking at it from the top, all you're going to see are those crossing overs. Does this make sense a little bit? Does it really? Or are you just saying that? Because if it doesn't make sense to you three, it's not going to make sense to them.
It makes sense.
Okay.
That's the one where you when you just the one you just showed us of the X-ray and it looks
Yes.
like the X. That's the top of it.
The X-ray is the top of it. This picture is what your actual strand is going to look like. These three are representing it. Does that make sense?
Yes.
Okay, cool. And so this picture indicates that the nitrogen bases at the center point inward and are equal lengths in binding because it's always one pyrimidine or one purine, right? Remember that pyrimidine is the long word, so it's CUT, which is cytosine, uracil, thymine. Purine is the short word, and that's going to be your adenine and your guanine. And so adenine and guanine are always longer base pairs, and your pyrimidines, your cytosine, uracil, and thymine are going to be your shorter ones. And so I remember this in a way that I don't want to tell you guys because I don't want you to get confused. But I'm going to tell you anyways. It's not scientifically sound. This is just a way to remember it, right? I'm trying to think of a rule for you guys, but if you don't do band, then I don't really know any rules. Like, I don't know. But the way I remember it is that your pyrimidines have been cut. Pyrimidines, I'll write that on the screen. Pyrimidines, golly, have been cut. C-U-T. C stands for cytosine, U stands for uracil, T stands for thymine. And they've been cut, so they're going to be short. Does that make sense? Mm-hmm. Whereas your purines are just adenine and guanine, so they haven't been cut, so they're still long. Does this make sense? Okay, that's not like a real thing. That's just the way that I remember who's long and who's short, right? So like when you guys go to take a test or when you take AP biology or when you're in college, don't tell the professor, yeah, pyrimidines were cut. No, that's just how you remember which base pairs it is and whether it's long or short. Does that make sense? Okay. And so purines, adenine, and guanine are larger nitrogenous bases, while pyrimidines, cytosine, uracil, when we're talking about RNA, and thymine are smaller nitrogenous bases. Please discuss what the links would be. So basically, what that means is that if they didn't pair up long and short, what would happen?
It'll mess up the DNA
Why would it mess it up?
because it's not paired with it.
They're not equally.
Because they're not going to be even sizes, right? So if I paired up short and short,
It's still gonna be short.
it's still going to be pretty short. And then I stacked it on top of a long with a long.
It's not
It's not
gonna be
gonna be equal.
It's not going to be even. It's not going to be that nice even double helix, right? It ended up looking something like
scrub.
that, which we don't want, right? Because does that look stable? Does that look like something you would want to climb up or something? No. And DNA is all of your genetic information. You want that to be stable. That's gotta have it together. If and your whole body has itself together, you need your DNA to have it together. And the large areas around the X are the sugar phosphate backbone. That's what we just talked about. Your sugar phosphate backbone is this area right here. I'm not very good about changing colors, is what I'm seeing here. But, so we've got our sugar, is this little blue guy? I know that was a brown marker. Don't even start with me. Our phosphate is this little yellow guy here. Right, and so we know those are the three parts to DNA. So it's going to be our phosphate, our sugar, which I'm going to do in brown again, and our base pairs. Right? Cool. Let's move on. Watson and Crick. So Watson and Crick get the most credit for DNA, which really all they did was like put the group project together, right? So imagine if everybody has their part. Like Julia's gonna do the introduction, Jemiah's gonna do the first paragraph, Kraisha's gonna do the second paragraph. We're gonna get Miss Hudnall to do the conclusion in the third paragraph, and then Miss Davis puts it all together. But my name's the only one that's gonna be on there.
That's not fair.
It's not fair, but that's how it goes. So all of these people played a role in figuring out DNA and understanding the way DNA works. But for whatever reason, most of the time, Watson and Crick are the only ones that get credit for it because of what they did, which is that they made the first accurate model of DNA.
It's right there.
So you got Rosalind, who's over here x-raying it. You got Chargroff, who's over here figuring out how all the base pairs go together. You got Hershey and Chase figuring out that there's DNA in everything. Everybody's doing all this important work. But Watson and Crick put it together, so Watson and Crick get the most credit, which I think is poop. Clearly, Jemiah thinks it's poop too, but that's just the way it is. So, base pairing and hydrogen bonding. So what actually happens is, like we've talked about before, is the bond between your base pairs is actually going to be just a little hydrogen bond, right? So guanine and cytosine are still going to hook up together in DNA, and adenine and thymine are still going to hook up together in DNA, but the actual bond is going to be a hydrogen bond. And that's what it tells you, Kylie. That's what it tells you down in that very last letter. Is letter E says hydrogen bonds hold the two sides together, and it is twisted into the double helix shape, so it looks like a twisted ladder. And you can kind of see where it looks like a ladder, right? Where each one of these would be a rung that you could climb up. Hydrogen bonds are weak bonds, and the reason for that is, I know we did it in second period. I don't know if we did it in first period. But remember that through here, you're going to have that enzyme that opens up the DNA so that way we can go in and make more copies of one side. And so if it was a really stiff bond, if it was a covalent bond, would it be easy to make more DNA? No, you'd have to have a ton of energy to break that covalent bond, right? So you want a weaker bond, because how often are we making more DNA?
Constantly until you have enough.
Constantly, not even until you have enough, but just constantly, right? Because we're constantly making more cells. You think about, how does your arm repair itself if you get a cut?
You gotta keep it. You gotta have it covered, and it's gonna close back up.
What happens with your cells, though?
Yeah, stitch it back.
They stitch it back together. You got to grow some new skin, right? That's why if you've ever gotten like a deep cut or a bad cut, what color is it when it comes back out?
You're not like a black color, or it's dark. It's a dark color.
Your new skin is black?
It's a scab.
That's the scab, but the new skin is pink, right? It's pink and it's shiny because it's brand new cells that have just been made to heal up that cut. Does that make sense? That's why most of the time babies come out lighter, don't they? Because they got all brand new, fresh cells. Does that make sense?
Yes.
Okay, so that's why we have these weak hydrogen bonds going through here, so that way we can break them apart, come in, make our new DNA, make our new base pairs, make our RNA, make whatever we got to make, and then we can close it back up real nice and easy. Because we don't want it to take a lot of energy. Otherwise, it would take a lot of work to make new DNA, right? How long does it take you to heal a cut? Like just a gentle, regular cut?
Like a paper cut?
Yeah, like a paper cut.
Like a week.
I'd say about a week, right? Can you imagine if it took like months to heal a paper cut?
No.
What happens if you really got hurt? then you'd be straight out of luck, huh? Yeah, so that's the whole point to those hydrogen bonds. We need to be able to make DNA whenever we need it. Oh, that was the end of that slide. All right, so moving right on into part 2. I think we pretty much covered everything on part 1. Let me double check.
Yes.
Okay, so from part 1, you need to make sure that you understand what everybody did. So we talked about a couple of different scientists there. And we said that at the end of the day, all Watson and Crick really did was put the thing together. But you need to understand who did what. So let's go ahead and look at part 2. Quit playing footsies with me. Let's look at part 2. And these notes are going to be due, what's today, Wednesday? Yeah. These notes are going to be due Friday morning. Does that make sense?
Yes.
Okay. So if you want to go ahead and write them down now, you can. If you want to turn off the video now and watch this video later, you can. It doesn't matter to me. Obviously, I'm not talking to you guys because you're in the room with me, so you're just going to have to sit through it. So what we're looking at here is S phase of the cell cycle. What is it called when cells divide and make 2 identical cells? It's not in there. You need to have been studying your notes. Keep on flipping back. Figure it out. I'll wait on you. Because there's a good chance if you 3 ladies need to look it up, the rest of your classmates need to look it up. What is it called when a cell divides into 2 and makes 2 identical cells? It's a type of cell division, but what kind of cell division?
I don't want to say that.
You better say something. Better call out all your names. No, telophase is a part of it, but what is that whole process called?
Mitosis.
Is it mitosis or meiosis?
Mitosis.
Mitosis is correct. Mitosis is going to make 2 identical daughter cells. Meiosis is what made you, right? Meiosis made me. Mitosis makes identical. So meiosis is where you got half the information from mom, half the information from dad, and it made 1 cell. Mitosis is it started with 1 cell and made 2 identical cells. Does this make sense?
Yes.
I'm going to give you superstars a hint here, and everyone else who dug around. Mitotic phase, mitosis.
Oh my God.
Oh, so it was on the screen.
I feel slow.
Come on, guys. So, DNA replication is the process of making a complete copy of an entire length of DNA. So this is just like regular mitosis, right? Because mitosis is the process of making a complete copy of a new cell. And so this occurs during the S phase. So, remember that we said S stands for synthesis. The word synthesis means to make. This should be in your prefixes and pearls. And so this occurs during the S phase of the cell cycle for mitosis. It'll also occur during the S phase of meiosis. Meiosis just has extra steps to it. That's really the only difference between mitosis and meiosis. And, you know, you get half your information from someone else and you don't in mitosis. Next. Complementary base pairing. What does that sound like? What is a complement?
Something nice.
Something nice. So if like I say, Julia, your sweatshirt really complements your eyes. What does that mean?
It brings out my eyes.
It brings out your eyes, right? It's like something that goes well with each other. So when we're talking about these base pairs, and it says complementary base pairs, what do you think that means?
They're coming together.
Base pairs that go well with each other, right? Can you tell me what complementary base pairs would be?
Adenine, thymine.
like adenine with thymine. And we see in this picture here exactly that, right? Adenine's got a point, kind of like, I don't know, when Miss Davis made you put a hand out. Oh, snap. Look at that, you were learning and you didn't even know it. And thymine's got this indent, right? Which is where we put a hand with a hand. Goodness, oh my gosh. Cytosine has this indent, like, I don't know, maybe a hand that's curved to catch a fist, which is like when Miss Davis made you link up fist with a, oh, wow.
God.
Wow, we've been learning this whole time and you didn't even know it. So that's all that complementary base pairing means, is that they're going to pair up together. So what you should notice here is that if I tried to put adenine with this pointy end with guanine, it wouldn't work, right? Just like if I tried to put adenine with cytosine, it still wouldn't work because it's not pointy. Does this make sense?
Yes, ma'am.
Okay, perfect. That's exactly what I want you to see. And it says it's easy for cells to do this DNA synthesis, this DNA replication, because they're going to line up together, right? It's going to be easy to put something together when all of the pieces have to match a certain really easy way. The semi-conservative model best explains the process of DNA replication. Over here yawning in my face, root. And it's not going to talk about that with you guys on the slide for some reason. Parent model, first step in replication is going to separate the strands. Okay, it does say, it says right there, duh. See how the dark blue strand is half of the new strand. And so the semi-conservative, semi means what? Half or like part of, right? Like, have you heard of semi-sweet chocolate? Like when you're baking, you have semi-sweet chocolate chips? All right, weirdos.
I know I've heard of a semicircle.
Yeah.
A semicircle, right, is like part of a circle. Does it always have to be half?
No.
No, it can be like a quarter, 3/4. It's just saying part of, right? So if it is semi-conservative, what does conservative mean? If you're conserving energy, what are you doing?
Saving it.
Saving something, right? So you're semi-conservative, you're saving part of it. And so that's what it's trying to show you here, is that when you make a new strand of DNA, you start with one, and you're going to split it open so that way when you make two, you saved what you originally started with instead of making a brand new one. Does this make sense?
Yes.
Does it really, or are we just saying that?
It makes sense.
Yeah, wait. This we're talking about this one?
Yes.
Right.
Yes.
Okay, you said it says part of what it already had to help make a new something.
Right, so we opened up this old guy, which is this dark blue. Let's go ahead and draw on there so people aren't confused. And we're going to do purple, because I like purple. So we started with this guy, right? And we opened him up into two pieces. So we see how these two are the same thing. One's just been spread apart. Does that make sense?
Yes.
Okay. And so then what we do is we come in with this new guy, which will make green. And we start adding in green pieces here and here. Does that make sense so far? So that way when we come out on the other side over here, we end up with the pieces that we started with and these new guys that we just added in over here. As compared to if we tried to match up green by itself and purple by itself. That wouldn't be conservative. Does that make sense?
What? You said if we added green and
So
purple.
this is conservative because it's half purple and it's half green. They're connected there, right? These two made two brand new guys instead of using any of the old pieces. So another way we could think about this is, let's say we're building a house, right? And so we can either build a brand new house from scratch Is that gonna save anything?
No.
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 More money.
Or we can use the pieces from this old house to build a new house. Did we save anything that way?
Yes.
Okay, that's the same thing that we're doing here. Instead of building brand new DNA, we're gonna use pieces from the old DNA to make a new DNA. Does that make sense?
Yeah.
Okay. I
Isn't
don't
that
think,
kind of like when you break your arm, like when you split your bone in half, you still have.
I've never broken my arm.
Oh, I have.
Oh,
Okay, you break your bone is like split, and then they still have both the pieces, and then they gotta make the new reconnect or something. What? Never mind.
okay. So Matthew Melselson and Franklin Stahl proposed this model in 58, and it shows the DNA side serving as a template or a guide for making the other DNA. And again, all we're doing is just matching up those pieces because they're complementary. Their application work is being done in opposite directions. Am I playing footsie as a someone? Okay. In opposite directions on both sides at the same times. In humans, it takes just a few hours to copy over 6 billion nucleotides. So that's 6 billion A's and T's and G's and C's. And it takes just a few hours.
It's a lot.
Just a few hours to put 6 billion things together?
It's kind of good.
That's really good.
A lot.
What can you put 6 billion of together
In
in a couple of hours?
Nothing.
Yeah.
Them whole
Yeah. Think about like how many sandwiches do you think you could make it? I will make it really easy, not even like peanut butter and jelly, like just ham and bread sandwiches.
Alright.
Do you think you can make 6 million of them in a couple
No.
hours?
I might make
Absolutely not. Absolutely. I don't think I can count to 6 million in a couple of
like
hours.
one piece of bread or two.
What?
Next.
Julia.
Do you eat bread? Do you eat a sandwich with one piece of bread?
I mean, like you can have a ham and a cheese and the bread and smush it over in half and then eat it like that.
Anyways.
Lord Jesus.
So the origins of replication. Origins is just going to mean starting point. Origin is just going to mean starting point. There's specific nucleotide sequences encoded in the DNA strands that act as starting points. And so it's showing you right here, you have an origin of replication or a starting point. The enzyme helicase, remember that ASE means what? Enzyme. Great job, guys. So helicase is going to be the enzyme that unwinds the DNA double helix, which kind of makes sense, right? Because helix, they sound about the same. So helicase is going to unwind it. It's responsible for unraveling your DNA. And what this does is it creates a replication bubble, which provides the spaces to do the actual building work of making the new complementary strand. So what we're doing is we're separating out. We have old DNA here, and we're gonna come in and we're gonna add new DNA right here. We're gonna add new DNA right there. And so, does it look like you only have one origin of replication here?
No.
No. How many do you see?
3.
3, right? Why do you think we might need 3?
Cause
What would that do?
Speed up the process.
It's going to speed up the process, right? It's going to make it go a whole lot faster as compared to, okay, I open this one spot. I'm going to go ahead and copy this. Now I'm going to close it. Now I'm going to move down. Is it going to be easier for me to make 6 million sandwiches by myself, or if we all start making sandwiches?
If
we all start.
If we all start making sandwiches. So these origin of replication, you have multiple replication bubbles. That way you can be making more DNA in separate places at the same time. So, DNA replication elongation. I think that's the next slide we have. Yep. So, elongation, it tells you, what does it sound like?
making it long.
It's making it longer, right? What is it making longer?
Well, no.
Look at your notes. What does it say? DNA, what?
Pop.
Complementary
Where do you
sizes
first see
requiring-
elongation?
Right here.
What word comes in front of elongation?
Replication.
What is elongation making longer?
DNA.
DNA replication. It's making that area to replicate longer. Right? So elongation of new DNA complementary side will require the enzyme polymerase. We got ASE in there, so we know it's an enzyme, even though it told us. It requires polymerase 3, DNA polymerase 3. This enzyme performs the addition of new nucleotides to the DNA complementary side and also acts as a proofreader to help prevent errors in construction from occurring. Look at the name and see the function. So remember that polymers were many units, right? Or many monomers, and we know that monomers are our building blocks. We should remember this. And we see that ACE tells us it's going to be an enzyme, so it's going to be an enzyme that adds in many units. You see how we figure that out? You need to be able to think like that without me walking you through it, because that's the kind of thing you're going to see on your tests. So, in this case, the monomers are called nucleotides. The ACE ending in polymerase tells you that it is an enzyme. So, I forgot what I was going to tell you, but I'm going to get there in a second. Yes.
What is this little zipper looking thing?
Oh,
said, like
I'm
a
so
zipper.
glad you said that. So, on the last slide, we had dark blue. DNA was our old DNA. So right now, let's change our colors. Okay. So on the last slide, we had these origins of replication. Do you remember seeing this?
Yes.
Okay. And then inside those origins of replication, you had these little things. Right?
Yeah.
What are these little things? Do you remember?
Those are the things that was helping make the new.
They're not the things helping make the new DNA. Those are the new DNA. Right? So with that in mind, what do you think this light blue area is?
The new DNA.
New DNA. And what about this one?
The new DNA.
New DNA, new DNA, new DNA. So I want you to, in your mind, go ahead and make note of which way that DNA, the new DNA, is traveling. So it's got arrows drawn on there for you. Those are going to be really important coming up. Like super duper important.
So it's after left to right.
Do they? Is that left to right here?
Like,
This?
no.
They jump from left to the right.
This one's traveling from the left to the right?
No.
From right to the left.
This one travels right to left. Does
Right
this
to
one
left
try?
and left to right.
Left to right.
So what you have is called a leading strand and a lagging strand, and those are important terms. Leading strand and a lagging strand. And so the one that goes from, we call it a 3' to the 5' is how they're always going to travel. The one that is really easy to make, straightforward 3 to 5, is your leading strand. Now, one of these, in order to make it, see, this is so confusing. I hate. Oh, awesome. It does tell you about your leading strand and your lagging strand. We'll talk about it again in a second, but your lagging strand is going to be made in chunks called Okazaki fragments. But so anyways, what this is showing you, now that we've gotten way off task here, you're okay. What this is showing you are your polymerases, which are these bubble things. That was the whole point to this slide. And so go back up to the top of Roman numeral 3. And it says your DNA polymerase 3. So what polymerase 3 is doing is you can see it's up here in the front, right? So it's unwinding this DNA. That way we can get in there and make our copies. Because when it's wound up like this, can you squeeze anything in there?
No.
Let me redo that with the point. When it's wound up like this, you can't squeeze anything in there. So you need that polymerase to come through and unzip it, which is why it looks like a zipper, or unwind it. That way you can get in there. Does that make sense? Okay, cool. And so it says this enzyme works at a rate of about 500 nucleotides being added per second. So every second we're adding in 500 adenine, thymine, guanine, cytosines, whatever the combination is there. DNA replication by adding nucleotides on the 3' end. So you have your new strand, you have your template strand, which is just another way of saying your old strand. The two sides of the double helix are said to be anti-parallel. What does parallel mean?
I mean, they're like this.
It means they line up like across from each other, right? That would be two parallel lines that we have right there. So if it's anti-parallel, what do you think that means?
They're not.
They're
like,
not-
side by side.
They're not
Hmm?
to each other.
We have another word for not side by side. We call that like perpendicular or diagonal, right? So it still has something to do with parallel. Otherwise, we wouldn't have said that word parallel, right? What your anti-parallel means is like one's going up, one's going down. Does that make sense? How they're still parallel, but now they're anti, right? So one's going up, one's going down. My
Like an
hands are
elevator.
still parallel. Yeah, like an elevator. One's going up, one's going down. They're still parallel because they're still on those same tracks, but they're going in different directions. This means the DNA information runs in different directions. Anti, but the sides are next to each other, so they're parallel. So if you three have been reading your notes, you could have read that already. DNA is always read and made 5 to 3. Remember that. I think I said 3 to 5 on the last one. But remember, it's always made 5 to 3. 5 prime, which is what that little quotation mark thing is called.
What is that saying?
My phone. That little doohickey is called a prime. Make sure you know that. And DNA is always read, and it's always made 5' to 3'. The 5' carbon of the sugar has a phosphate attached to it. The 1' of the sugar has the nitrogenous base, and the 3' carbon of the sugar has an open bond, which is the connector site. Helicase enzyme causes the double helix to unwind. Let's see if they have a side. Oh, cool. They have a picture showing you those primes. Look at that. So your 5' is this guy right here. And like we just said, 5' is going to have a phosphate. Your 1', which is right here, is where you're going to have your base pair. So this is where you're going to have adenine or thymine or guanine or cytosine coming off of it. And then your 3' has an open bond, right? And so that's going to be where it connects to the next piece in the chain. Does that make sense? I hope so. So your lead strand and your lagging strand. Helicase causes the enzyme double helix to unwind. We already kind of talked about that. Single strand binding protein keeps the two sides apart and stable. Why is that important? Why do we need a protein to do that?
So it can function right.
So it can function right. What is it?
The cytosine.
We're not talking about cytosine necessarily right here. What I want you to think about is what kind of bond holds our nucleotides together? What holds our base pairs together? It's in your notes. We've been talking about it.
It's an A and a T word.
What that mean?
The thymine.
Is a thymine a type of bond that we've talked about?
Oh, you said the it's all let's see. You said a hydrogen bond and then a covalent. Didn't you say covalent?
So which one holds our base pairs together?
The covalent bond.
Covalent bond holds our base pairs together. Now, if a covalent bond held our base pairs together, would it be easy or hard to make new DNA? Hard, because covalent bond is the strongest bond we have. But we just saw that in order to make new DNA, what we have to do?
Make it easy.
You had to break the bond, right? So if we were constantly having to break covalent bonds, it would take a lot of energy, wouldn't it?
Yes.
Does it take a lot of energy for you to heal a paper cut? Are you like, oh God, I better rest for the next week
No.
while my body heals? No, that's something that's done pretty easily. So do we think that it's going to be a covalent bond that holds our base pairs together?
No, it's gonna be a
No.
hydrogen.
It's going to be a hydrogen bond. Now, what did we say about hydrogen bonds? Do you remember that day where we had the middle of the classroom before we brought all these tables in? And I ran around trying to break all the hydrogen bonds. What happened as soon as I broke one?
You broke them and they came back together.
They came right back together. So if DNA helicase is doing all this work to break the hydrogen bonds and they're just coming back together, would we have time to make new base pairs? Will we have time to add anything in? No, because it just snapped back together and wind itself back up. So that is the whole purpose to that single strand binding protein, is to keep the sides apart. So that's a really important thing that I would know, but you guys do what you want, since I see you just sitting here looking at me. Your leading strand of the replication fork. So the replication fork is this whole thing. Do you see how it kind of looks like a fork? Right here,
Yes.
right? So you have the leading strand, which I wish I could just tell you, no, it's the top strand or the bottom strand. But your leading strand makes it very easy for us to move from 3' to 5'. So if you're looking at just the dark blue, you see that your 5' end on the top strand is over here. Your 3' end is over here. If we're doing DNA replication, and it's always read and always made from 5' to 3', do you see how it's real easy to just go choo 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 choo? As compared to over here, right? Because they're anti-parallel, right? Because I feel like I lost you there. So if it's 5' to 3', you would read this top strand from left to right. Does that make sense? But because replication is anti-parallel, you do it the opposite direction. Does that make sense?
Yes.
So when we're making the new one, we're making it from its 3' end, which is right here, to 5' end. So 5 to 3. Does that make sense? It's made from 5 to 3. Do we get that? Are you sure that makes sense? Okay, I hope it does. So it's real easy to just go in and fix these pieces in, and we don't have chunks. As compared to over here where it's 5 to 3 going the opposite direction, which means that when we make it, it has to go 5 to 3. Right? Because it's always made from 5 to 3. So you can start right here and you make a 5 to 3. Then you got to stop and make a new one, 5 to 3. Stop, make a new one, 5 to 3. Because can you start over here and just keep going?
No,
Why not?
because it's because that's because the one we have to do 5 to 3, that's the one we can just go straight.
Everything is 5 to 3. It's always 5 to 3.
I thought this one was 3 to 5.
Look at your notes. What does it say? It's always 5 to 3. The problem is what happens when you get right here.
Because it's starting the lagging, the lagging strand, and it's not focused on what it's doing. And
It is focused on what it's doing. It's very focused on what it's doing. Think about it as I really don't know a good example here. But the issue is that this area hasn't been unraveled yet, right? Yeah. So these little bubbles are our polymerase, and they're working to unravel it. Now I have to go 5 to 3. There's no ifs, ands, or buts. Can I start over here if it hasn't been unraveled?
No.
No, I have to start where it's opened at. So I can do 5 to 3 right here, and I can keep on going forever and ever. The problem is that our origin of replication is a bubble, right? So if I start right here, I can only go until that bubble closes. Does that make sense? And then I have to stop. Now for this guy, for our leading strand, he can just keep going as polymerase keeps opening. He doesn't have to stop because that's 5 to 3 for him. But for this one, I have to wait for polymerase to open it. And then I have to start over and wait for polymerase to open. And then I'm going to start over and wait for polymerase to open. Does that make sense?
Yes.
Okay. Let me see what your notes say. Your leading strand of the replication fork goes 5 to 3 and is continuous. So go ahead, underline, highlight, circle, continuous. To start adding nucleotides, you first have to attach an RNA primer using primase, right? That should be a really fun, easy enzyme for you to remember. Primase. Ace means it's an enzyme. And it says prime, so you know it's going to be priming something. It's getting something ready to go. And so this is talking about the DNA being made, not that side you started with. Remember, it's semi-conservative, right? So you've got the start that you already had, your template, which is this dark blue, and then you've got this new side. So the primase and RNA primer are going to be working with your new DNA, not your old DNA, right? Because do we need to add anything to this DNA?
No.
No, it's already got all of its information. Your lagging strand, this side of the replication fork has DNA not running in a 5 to 3 prime direction. Therefore, it's always lagging behind because it's having to stop and start and then move over and stop and start and then move over and stop and start. Does that make sense? Mm-hmm. Okay. Well, we'll talk about it in a second. This side of the fork has to wait for the long segment of DNA to become exposed first before it can start adding a primer. When a long segment has been opened, an RNA primer will attach, and then DNA polymerase will work backwards to create an Okazaki fragment. And all an Okazaki fragment is that chunk of DNA, because we had to start and stop and start and stop and start and stop. The Okazaki fragments are stitched together using the enzyme ligase. So let's add in a slide right here.
Ligase is an enzyme, right?
You tell me, what does it end in?
ASE.
So what does that mean?
It's an enzyme.
So what happens is we've got these two pieces of DNA, right, in this double helix. And so we've already decided when it's all wound up like this, we can't get in there. So what happens get out of my way, little weird thing. There it goes. No. Is you have that polymerase. Come on. Thank you. You have that polymerase that'll come through and it says open up. Does this make sense so far? Yes. Okay. And so once it's opened up, then you can have your new DNA start to be made. Now, the thing to keep in mind is that you still have DNA over here that has not been opened, right? And so if I'm going from 5' to 3', which I have to, and I'm going this way, then it's very easy for me to keep drawing that blue line as this polymerase keeps opening, right? Whereas with this one, I have to go the opposite direction because our strands are anti-parallel. So they're running in opposite directions. So let's say that I do it like this, right? We'll say that whole chunk is nice and easy to make. We all good so far?
Yes.
Okay. Now what happens is, do we want to leave this DNA just open, hanging out there?
No, you want to close it back.
No. So it starts to get closed back. So over here, once polymerase gets out of the way, we're going to open up a new segment of DNA. And we're going to close off that old segment. Now, for our leading strand, this isn't a problem, right? Because that new DNA or whatever we're making can just keep hanging out the backside. Do you see that? So what we made right here is now hanging out up here. Does that make sense?
Yeah.
As compared to over here with our lagging strand, it can't hang off, right? Because we're doing like this. I can't jump on and add more here. I got to start a new one like that. Does this make sense?
Yes.
Okay. So the next time that we open up our DNA, sounds like gunfire. We still have on our leading strand, now we've got this long, huge, spirally piece of DNA. Whereas over here, we've got our chunks on our lagging strand where we have to keep coming back in and starting over. Does this make sense? Do you understand now why we have a leading strand and a lagging strand?
Yes.
It's not that our lagging strand's being lazy or is dumb or doesn't work as well or anything like that. It's just that it's having to go back and jump over itself, right? So we made this piece first, then we made this piece, then we made this piece, then we had to make this piece. As compared to, we made this one, this one, this one, this one, this one, this one, this one. Make sense?
Yes.
Cool. RNA primer is our starting point, so it's just gonna prime, it's just gonna get things ready for us to go, for us to start that replication. Your nucleotide excision repair. Okay. Where have we heard excision before? Where have we heard EX? What? At the doctor. Think about in class. Exits, right? EX exits. So excision, what do you think that's talking about? Something's leaving, right? What comes in front of it?
Nucleotide.
Nucleotide excision repair. So what do you think that means?
Nucleotide is leaving
That you're taking out a nucleotide. Why?
to repair it.
To repair something. Exactly right. So what it's showing you here is that for whatever reason, the thymine dimer distorts. So you see how it's got this bump, right?
Yeah.
So what we're going to do is we're going to say, fine, you know what? You want to act up? We're going to take out this whole section. Now, think about your DNA as back to being your house, right? Mm-hmm. Can you just go in and take out chunks of your house when they don't want to act right?
No.
No, it's not a very stable house, is it? Right?
Okay.
What is your DNA?
My DNA.
Yeah. What does it do?
It holds all my genetic
It
information.
holds all your genetic information. Do you want that to be unstable?
No.
No. So what happens, even though we took out this broken piece, we can't just leave it with a big empty hole. So what happens is we start to fill in. So we're still going to work 5' to 3'. But now we're going to build in a new place. That way our DNA is still good to go. Does this make sense?
Yes.
Now, did I just make up some new information here?
No, that's the exact same
same
thing as
thing.
the other one that we just looked at from the other.
Because it's still going to match up with what we already have here, right? Whether it's a curved end, whether it's a pointy end. So we know adenine has to go with thymine. Guanine has to go with cytosine. It doesn't matter how it feels that day. It doesn't matter if they're in a fight. They're going together. Does that make sense?
Yes.
Okay. And this is a correction of errors or proofreading. So this function is performed by polymerase 3, DNA polymerase 3. which is the same one that's opening things up. So mismatch repair is when the wrong nucleotide is added to the new sequence. So we've got a couple of different types of pairs. And your DNA polymerase will reverse. What does reverse mean? Go back to a spot, remove the wrong nucleotide, and then replace this with the correct nucleotide. So this is like if I misspell a word on my Facebook status post and I hit what button to go back?
No, it's like a little
I just
thing.
hit the backspace.
I thought you were talking about when you already posted
I haven't posted it yet. I just hit backspace and type over what I want, right? That's your mismatch repair. Now, it tells you that you also have, for errors that are created after your DNA has been after your DNA has already been made. So now I've already posted that Facebook status. You guys might not have needed to turn your page. Mine's different. I've already made that Facebook status. Now I need to go back and fix it. That's when you have your excision, right? So that's where I'm going to go in. I'm going to delete that status or whatever was wrong with it, and I'm going to put in the new status. Does that make sense?
Yes.
Okay. And so the way this works, step one, your nuclease is going to cut out this part. So step one is right there. Step two, your polymerase is going to add in your new nucleotides. And step three, your ligase is going to attach your pieces back together. Does this make sense?
Yes.
So I guess it's like glue, kind of.
Your ligase is just like glue. And so errors in proofreading can result in some forms of cancer. So this ability to repair itself is like another checkpoint. It's not an official checkpoint, but it's like another checkpoint. So for instance, it tells you some individuals are genetically predisposed to skin cancer because they have a mutation in the gene that codes for the excision repair enzyme. They can't fix damage caused by UV lights on skin cells. So the fact that their enzymes can't do this excision repair means that you get a bunch of these faulty cells. You get a bunch of bad cells. Now what happens when you have a bunch of bad cells? What do?
They die eventually, don't they?
Well, they die eventually, but what do they do before they die?
They make more bad.
They make more bad cells. So I might have started out with 100 bad cells. What's going to happen the first time they replicate?
You're going to get 200.
I'm going to have 200 bad cells. What happens when all 200 of those replicate?
I'll have 400.
400. What happens when they replicate?
500. Wait, they're going to die.
So 100 cells
Uh-huh.
split into two.
Uh-huh.
It made how many?
200.
Split into two. It made how many?
400.
Split into two.
800.
800. Split into two.
16.
Split into two.
60,000.
1600. Split into two.
18.
A million.
3200. Right? 3200. 16 times 2 is what?
Oh, yeah.
Okay. So now if they're doing all this, how long does it take for you to build cells? I've been talking about
Like good cells or bad cells?
any cell. How long does it take for you to go through this whole cycle?
A minute.
A couple of minutes, right? So in a couple of minutes, we'll say maybe an hour. Maybe your cells are really slow this day. In an hour, I've gone from 100 bad cells to 3,200 bad cells. You see how cancer can come on really quick?
Yes.
Okay. So that's the end of these notes. Big things I want you to remember are Chargaff's rule, which are going to tell you what? What was Chargaff's rule about?
Adenine equals thymine, and cytosine equals guanine, or something like that.
Tell me in your own words. What does that mean?
That means you gotta make them equal, right?
Make what equal?
Make the guanine, the adenine, the...
What are those things called? What are those 4 letters called?
Organ? No.
Look at your notes.
Say what it is called.
Mm-hmm. you guys need to be studying your notes on macromolecules. That's the last time I'm going to tell you guys. Those are your base pairs. Chargaff's rule just says that your base pairs always match up to the correct base pair. It doesn't matter where you and Bay go on a date, you're always going to be with Bay. A always equals T, G always equals C. And A plus T plus G plus C always equals 100%. Does that make sense?
Yes.
Okay. So Chargaff's rule is just saying that A and T are always going to match up, G and C are always going to match up. And they're always, all four of them together are always going to equal 100. And that's what you need to take away from these notes. So I hope they helped. And I will see you guys tomorrow.